Okay. So we talked a little bit about chromatin remodeling complexes, and this sort of shows you a little more about how they are and how it's all linked together. So you notice here's this particular piece of DNA. It's in that 300 angstrom fiber. It's the nucleosomes are associated with it. And some of the DNA in there has the tau binding. So the tau binding site is wrapped around that nucleosome with this gene that has to be expressed. In order to express it, several things have to happen. So first of all, here's a gene activator protein, right? And it's bound, it's got a piece of DNA we can bind to. And what these gene activator proteins, so these are proteins that bind around the promoter region like that SP1 and so forth. And the one thing that they can do is they can attract these chromatin remodeling complexes, CRCs. And that chromatin remodeling complex, right, interacts with that DNA and it can change it, like I said. You can see in this cartoon, they've changed the spacing. They've changed the number of nucleosomes that there are. There would normally be a lot more of them there. So the DNA is actually remodeled. These guys can change that. One reason that they do, or the other thing that they can do, are these molecules that are called histone chaperones. And histone chaperones are actually can bind to the histone, to the nucleosome core. You can see here there are regions of the DNA where the yellow's going, because these chaperones have actually removed nucleosomes. Remember I told you they can remove them. And notice, all the time, notice where that tau box is, right? That tau box now is in DNA that's sort of becoming exposed. So as it becomes exposed like that, then the cell can have proteins that bind, like the transcription factors and so forth, to begin transcription. So this is whole nucleosomes removed. Another thing we can do, the histone chaperones can do, is they can remove part of a nucleosome or exchange. Remember I told you they can put in a special histone. So here you can see they've exchanged for a special histone. And usually what those specific histones do is they help to unwrap or loosen that DNA. And then finally, here's the enzymes we talked about, histone modifying enzymes. Often they bind to these gene activator proteins, or they are gene activator proteins, right? So those histone modifying enzyme are these HAT enzymes or HDACs. In this case, the HAT enzyme is acetylating this DNA. And so you modify these histones within this region, just once again, to loosen transcription. So all of these things are working together. Right? You can see even in this cartoon, they sort of have them drawn separately. This histone activating thing, these enzymes here, are drawn separately from these three different types of remodeling that the remodeling complexes do. And the remodeling complex, it depends on which proteins are associated with it. You may have heard of SWI-SNF, SW1 SNF complex. That's a chromatin remodeling complex, one type. It's found in this particular cell. But all of these lead to transcription, right? And you can see that as this is loosening up, the general transcription factors, those are TF2s, and mediator ends, which we didn't talk about, and RNA polymerase 2 are all binding in this region, in this promoter region, to begin transcription. Have a question? Okay. So this is just, once again, showing you a little more about what these remodeling complexes can do. And if you go through and you look at these individuals, you'll see here's the standard nucleosomes tightening. One thing that the chromatin remodeling complex, as we said, they can cause these things to be loosened. Right? They can also do this, which is called phasing. You can see the distance in between is different, so they cause them to move a little bit. And then here's a remodeled nucleosome. These guys that are written. So SWI-SNF complex is a complex that can rearrange DNA in nucleosomes so the transcription factors can bind. That's just one example of these. Okay. So in this, what we're talking here about these chromatin remodeling complexes actually making changes here. This is the kind of changes that are made. Okay. So here we're back to this eukaryotic proximal promoter region we talked about, a couple hundred nucleotides. Here are the four major sites, the TATA, the GC, the CAT, and the octamer. And all of these are regions that bind specific proteins. So these are sequences in the DNA, right? Those are the DNA sequences there that are consistent. But there are proteins that bind to those that cause things to happen in the see the TATA controls the choice of transcription start point, so that when the transcription factors bind there, the TATA binding protein, because the spacing in this region is uniform, it causes transcription to start at +1, not at -3 or +3 or whatever. These regions here control initial binding of RNA polymerase. So particularly the GC box, the CAP box, and these two really are the ones that regulate binding, initial binding of polymerases. Okay. We talked about these. I'm not going to talk about them again. So I want to talk a little bit about enhancer elements now. Enhancers, we talked about proximal promoter regulation, regulation of gene expression by proximal promoter regions to undercell nucleotides. There are other elements that we call enhancer elements, and it was because what they were first described as. They were described as regions that enhance the expression of a particular gene. We now know that those regions can be their gene activators or repressors. They can be either way. What's typical about these enhancers, or what's unusual, is that they're typical among all of them. There are several properties. One is that they can be very far away. They can be very distal. Some of these can be 50,000 nucleotides away from the gene itself. So they can be very far upstream, as this does. They also are able, you can take an enhancer and flip it 180 degrees in the DNA. It works just fine. It doesn't seem to, there's no problem whatever. And it has to do because once you bind a protein, as long as it's recognized, the sequence is recognized by the DNA binding protein, that protein then can then interact with the promoter in the gene to help to regulate expression. Not only can it be far away, but it doesn't have to be, the proximal promoters here is always on the 5' end of the gene, proximal promoter. The enhancers can be anywhere. They can be on the 3' end of the gene. They can be way down beyond the gene. They can also, in the case of ribosomal RNA, the 5S ribosomal RNA gene, there's actually an enhancer right in the middle of one of the coding regions for the gene. But these enhancer regions, remember, bind proteins. And when they bind proteins, they're able to interact with the proteins and the polymerase at this proximal region to enhance binding of polymerase. So usually the proteins that bind to these enhancer regions also interact with proteins, right, with the polymerase. And they actually help to load the polymerase onto the promoter regions. So generally, and certainly an enhancer will enhance the binding of polymerase to the promoter region. The other thing that this doesn't show is enhancers can work synergistically. They can be in tandem. The virus, SV40, has two identical enhancers, so they're duplicated. They bind twice as much protein, and they're extremely active activators. Okay? So why do we have enhancer elements? Do they just increase the rate of expression? They do. But the other thing is that the enhancer elements are often tissue specific. So there's an enhancer element that is where the protein that binds to it is in muscle cells. And that enhancer element is associated with genes that encode proteins in a muscle cell. Actin, myosin, tropomyosin, troponin, they all have that same sort of enhancer binding site. Well, of course, all the cells have that same enhancer binding site. Your DNA and all your cells are the same. But only the muscle cells make the protein, the enhancer protein, the activator protein that binds to that enhancer in muscle cells and causes the expression of that protein. Red cells don't make it, liver cells don't make it, kidney cells don't make it. So tissue specificity of expression is determined primarily by enhancer elements. So that means that you could recombinantly, you could take, perhaps you could identify one of the enhancers in muscle cells is called myo1. And if you take that enhancer and put it in front of actin in that cell, or put it in front of something, that's not a good question thing. If you put it in front of a gene that's normally not expressed in muscle cells, you can turn it on by putting that myo enhancer in front of it. Okay? Question? Okay, so this is just a picture of a particular gene. And this is a beta-globin gene. And you notice that here's the gene that runs from here to here. We're going to talk about exons processed and so forth. But if you look at that, that's the coding region of this gene, at least the white regions are the coding regions. And this is the upstream promoter region. And there's also a downstream promoter region in the globin genes. And you notice that here's the top box, and then here's all of these other proteins that are binding there. And all of these proteins are binding to different sites on this gene, and they all regulate expression of that gene. Some transcription factors, CP1, have elements in most gene regulatory regions. Okay, so if you look at the promoters on many, many different genes, most of them have one that binds CP1, which is happens to be this guy here. But there's a CP1 binding site. Others, this GATA1 protein, are found only in a very few cell types. So GATA1 is one of these, this green guy, is one of these enhancer elements that works in a tissue-specific way. It ensures that only red blood cells make globin and not muscle and not other. So the the protein that binds, GATA1, is made in red blood cells, but it's not made in muscle cells or liver cells. Does that make sense? So these proteins that bind to these sites, right, then recruit more chromatin modeling complexes, the histone modification enzymes, general transcription factors, and polymerases. So the binding of these proteins to the promoters activate transcription and bring in other proteins that are needed for transcription. Okay, so these figures are all in your book, and you should be able to take them. So quiz on 5.